I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
And we're giving everyone a pop quiz. Can you spell ophthalmology?
O-P-T-H-T-A... Wait, sorry.
No, I don't even know what that is.
O-P-T-H.
It's like O-P-T-H-A-M-O-L-O-G-Y.
That
No?
was the fastest anyone has spelled it.
Was it right?
I don't know, but it was fast.
Wrong, yeah. Okay.
O-P-H-A-L-M-O-L-O-G-Y.
Are y'all all in Opta?
I'm the one that discovered it was spelled wrong in the database.
Hi, everyone. So this season, we're concentrating on groundbreaking discoveries. And since discovery starts with vision, we're going to take a deep dive into what's really pushing the boundaries in ophthalmology. Back in season 6, we explored where vision research is focused. See what I did there? But today, we're going to learn about the wild ways that research is pushing for transformative therapeutic strategies. I'm excited to introduce and hang out with cutting-edge scientists who live and breathe vision. Susie Crowell, a senior director and project team leader, as well as an old friend, and Deepak Lamba, director and distinguished scientist. Welcome to the bar.
Thanks for having us.
Thank you.
So, like in our last eye episode, we like to start off with basic understanding. In that one, we covered how does the eye actually work in the biology, but I thought it'd be fun to get meta with it. Susie, how do we even perceive vision to begin with?
It's so much wackier than I ever thought about before I started working in this space.
Tell me more.
How eyeballs work is just wild. Basically all of our senses are just like taking whatever it is in our world and turning it into electrical impulses that our brain deciphers. And we're just so good at pattern recognition that we're like, hey, what if we paint it purple? Or whatever it happens to be. And it's a really complicated sense where we're taking light in through, the window, the pupil in the front of our eye. It hits our retina and then gets, transmitted through the optic nerve coax cable into the visual cortex in the brain, where the brain's like, I'm going to make meaning out of this. By like the number of things that have to happen there, it's really complicated. And it's kind of crazy when we talk about trying to make medicine that addresses things in the visual realm, because like, how do you measure if it works when we could have like a really bizarre philosophical discussion about whether I see the same thing that you see?
Yeah, it's complicated. It's not like light goes in, reaches the brain, and you see something.
Yeah.
There's different ranges of light. In fact, our light sensing cells, the light sensing photoreceptor cells, these cells have a weird way. So they're normally active all the time. So when they sense light, they stop responding to light, and that's how our brain senses. It's called hyperpolarization. So when light hits these light sensing cells, suddenly the channels close down, and that causes our secondary order neurons to sense light.
Right, so that makes me think of glaucoma, right? Where there's a problem with these neurons even communicating to begin with. How do we even deal with that? How do we get them to start working again?
The glaucoma is way more challenging because then that's the one and only connection between your eye and your brain.
It's like the cord is gone.
So you can't just join the frayed ends together. You actually have to have the new cells make a new cord all the way. And that is the reason it's not been very successful so far, because the cues that are there in the microenvironment to tell you, okay, this is your route to the brain, go away once you're fully developed.
What about for other parts of the eye that are needed for vision? Are there other places where there is damage that actually leads to pathology?
Yes. And there are different sort of initiators of damage which can cause different diseases. So like there are people who have inherited degenerations, for example, people like who have retinitis pigmentosa, which is this broad disorder where different mutations in different genes which are involved in light sensing are affected. And so that's the cause. So that causes abnormal proteins to accumulate inside the cell. And the cell's like, I'm done, I can't deal with it. They start dying. And so you lose those cells, at least early in the disease, the rest of the circuit's still there. Because it's a local circuit. Like the only one cell right next to it is gone. So potentially you may be able to replace it. But the big challenge with all of it is our brain and our eyes are so smart that they feed in the missing information. So essentially you can lose close to 80% of your cells and not realize you have vision loss.
That's super scary.
Because your
It is
brain's like
insane.
photoshopping it for you.
Exactly.
This is my thing because I'm missing like a huge chunk. So we're sitting in a triangle right now. We're all about 3 feet from each other. When I'm looking at you, Danielle, I can't see Deepak at all, even though he's in a part of my peripheral vision that most people would be able to. But there's just like a weird, navy blue smear. Like it's like an AI generative whatever. And it's my brain trying to be like, well, I know this was there a minute ago when she looked that way, so I guess I'm going to fill it in.
Yeah. The brain fits in the information but often fakes it. And so there are people who have advanced diseases where they don't realize it, but then they hit a pole and then it's like, how did I miss that? It was right in front.
Well, and then for some of these that are degenerating slowly over time, perceiving that change without having kind of, tests or something that's making you notice it is really difficult. So when I first started in this space, I thought a lot about neovascular age-related macular degeneration, where blood vessels are growing in parts of your retina they're not supposed to. And that causes leak of fluid, and the fluid, messes up the optics, essentially. Can't see. And there was a lot of talk about how patients don't comply with their treatments.
Yeah.
And if you're also, elderly and have somewhat impaired vision, it's not like you can just drive yourself there. It's a big problem. This isn't compliance. I would get very upset. Because my experience of losing vision slowly versus abruptly, I have experience with both of those. And it's a completely different situation. The slow loss, I only know about it because I'm obsessed with my visual field and I check it all the time.
Hey, Danielle.
Hey, Karen.
Hey, Danielle.
Hey, Wellington.
Danielle, how do you explain why compliance is so important for people who have eye diseases and are taking treatments? How is the field addressing this issue?
Oh man, that's like a huge area. So first of all, for compliance, the medications only work if they're at the right concentration in the target area for the right amount of time. So if you want to get to a certain concentration, you have to have multiple routes of administration, but it's like really uncomfortable, and the patient population that has to get it is already at a disadvantage because they can't see well, so they need to get someone to take them to their appointments. They're usually an higher age population, so there's a lot of hurdles to actually getting treatment. So to address that, there's a lot of work that's done to try to do what are called like long-acting delivery systems, or something that helps it so that you don't have to keep giving as many doses, and if you don't have to do that as often, it makes it easier for people to actually follow through. That's exactly what Susie and I worked on together. Most of the time, whenever I talk to folks about diseases in the eye, the most common culprit is problems with vascularization and VEGF. Deepak, could you tell us a little bit more about this?
So basically in some of these vascular diseases, what happens is there's a cue from the dying cells, which tells them we need more food. And that cue to the vascular cells is VEGF. VEGF stands for vascular endothelial growth factor. And so this factor tells them, oh, we need more blood vessels to grow in because we need more food. Unfortunately, it reaches a point where these cells are not controlled anymore, and so they just start migrating and invading into the eye. And then become leaky because they are new vessels. The body's not used to having new blood vessels in older ages, and so what our therapies are is to try to block that, to see if we can sop up all of that extra VEGF in that milieu so that your vessels now stop growing in, and that sucks up all the extra fluid.
So like a VEGF therapy is really something to sop up all the
Pretty
VEGF.
much.
Because it's like the cells are like, I'm hungry. Your body's like, panic. And it overbuilds and freaks out and it's leaky. And then we want to calm it down.
Damp it down. And so that affects that part. But again, as you can imagine, also part of that is if you're doing that and your cells are growing into the wrong spaces, leaking, that drives inflammation. And so those are the things that are missing right now. So we can target the VEGF really well, the vascular supply. What you are missing is what's around it. You're trying to tamp down the inflammation that's happening in that space. Also, these new blood vessels cause other defects which cause cells to become more fibrotic. And so that's another signal that develops in these patients where there's local fibrosis. And if there's a thick scar in the back of the eye, then the eye is not going to function well. And so those are the areas that we need to still try to figure out how do we get to those biologies. And these are all side effects, right? The main part of the disease is still happening that we still need to get to.
I think it's been about 20 years since anti-VEGFs really came onto the market in full form. And that was a massive game changer, as I think you guys have previously discussed, where patients weren't just, we weren't just slowing disease down. They actually got vision back as we closed down the new blood vessel factory.
Yeah.
And that fluid would go away. And that's been huge. And I think there's been a lot of iteration on the anti-VEGFs in the field. There's been a little bit of new biology introduced in this neovascular AMD and then the other kind of leaky diseases, so diabetic eye diseases. But there hasn't been another one of those massive transformative inflection points since then. There's been kind of chipping away.
Well, that's like the alley-oop for y'all, right? VEGF is for the vascular-related kind of pathologies. But what about the ones that y'all were talking about where photoreceptors have been damaged and are missing? Are those going to be in pathologies completely independent of those VEGF? Or how would you even address those? Maybe is a better question.
Both glaucoma and geographic atrophy.
Yeah. So basically, even when talking about just neovascular AMD, macular degeneration, that's the end stage of the disease. What leads to it is some of the earlier stages, intermediate AMD, and that can either lead to that or to GA, which is geographic atrophy. So which is the area is dying, the whole geographic area has atrophied. And that's where you're losing these light-sensing photoreceptor cells, the support cells at the back of the eye, called the pigment epithelium. And so there are lots of efforts on seeing, once you've lost these cells, can you replace them back? So there's a few things in the clinical trial stage right now of trying to replace either the pigment epithelial cells or photoreceptor cells. It's very, very challenging, as you can imagine, putting new cells in an environment which is undergoing active disease. So that's one area. The other area is, can we save what's being lost? Can we go in early, try to hit some of those pathologies early? So that's that. And then on the other side is glaucoma. Glaucoma is a disease where, as I mentioned earlier, the main cell going from the eye to the brain, the main connection, those cells are dying. Currently, all the treatments are to try to reduce the pressure. So often this is connected to pressure in the front of the eye. And people have lots of drops and things like that for it, but they never get to the main problem of the disease, which is the cells dying at the back. And so that's another big area where we need to figure out how to start saving them.
Maria was super excited that we were going to talk to y'all, and she really wanted me to ask you to give a little bit more detail about how cell therapies are actually going to potentially play a role in this area. Because, this is such a new place to be thinking about it. Normally for cell therapies, I think most of us hear about it in the oncology space, but not in ophtha.
There's some really cool stuff going on here and a lot of different approaches. So Deepak's mentioned a number of cell types that are at play here. You've got your nerve fiber that goes to the brain. You have the pigmented epithelial cells. I think of those as the garbage dump and grocery store workers in your eye. They're taking care of the photoreceptor layer, which is the business center. And then, there's a few other cell types in there as well, but let's focus on maybe those three. There's some cell therapy approaches where they're basically like, all right, in geographic atrophy, where a lot of those pigmented epithelial cells have dropped out and you just have a big desert, food desert. They're like, okay, let's just put a slurry of, a mix of RPE cells in there, fresh ones, and see if they'll, take root and hang out. So that's one approach. There's another where they're like, okay, let's culture, a film of RPE, so retinal pigmented epithelial cells, and then put the entire, like a fancy Band-Aid, just put that whole thing in there and
almost
unfurl
like a
it.
tissue graft kind of?
Yeah.
Dope.
Yeah, it's like really cool.
Yeah.
And then there's other ideas where it's like, all right, you lose your RPE layer, and what happens when you have a food desert? People move away. The photoreceptors, actually in this case, they die, so it's a little bit darker. But so then you have two problems. You need to reestablish the food and garbage capacity, and then you need the people to come back, the photoreceptors in this case. Mm-hmm. So let's put a mix of both in there and see if they work themselves out. And will they do that if you mix them together into a cell replacement smoothie? Or is it you have to do one first? Deepak's like, no, Susie. Do you do one layer first and then the other? Or do you need to do the tissue graft? There's a lot of geometry and architecture and gross smoothies, I guess.
Cell smoothies are super
so
popular
gross.
right now. I know.
I regret saying
that. But everyone and their mom is all about cell smoothies and
it's
seeing
crazy.
how
do these guys go where they're
other?
supposed to?
So everything she said is exactly how it is. People are trying to figure out how you can put either a single cell layer, the pigment epithelial cells, those grocery/garbage, which is very complicated to put it together, but that's exactly what they do. They are the main cells, because your light sensing cells actually have no vascular supply. So they actually rely on these pigment epithelial cells.
When they're so busy. These are the most metabolically active cells in your body.
I didn't even think about it, and I don't know how many retinas I've stared at, but yeah.
So they actually have to bring in the food, get rid of all the dead stuff. And what makes it even more complicated is our light sensing photoreceptors have this weird adaptation at the tip called their outer segment, and they shed 10% of that every night. Every night when you go to sleep.
Did you know that?
I did technically know that, but I don't really get it,
Why have we not talked about that before?
It's just it's a crazy system.
It's insane. So at night they just have to go crazy because they have to chew it all up, package it all together, and get rid of what needs to be gotten rid of, but also put things back so the photoreceptors can keep making more.
Yeah.
Because they rely on the same cells.
Little sidebar. The other thing that happens while you're sleeping is that the rest of your brain's the visual cortex isn't busy. What if we just put that to work for other things? So your brain's fighting to take that space away and give it to other senses.
Temporarily hide.
No, it wants to take it forever, and then you wake up and it's never mind. Let's give it back.
Shut up.
It's crazy. It's crazy. The whole thing is nuts. I can't believe it works.
Okay, Danielle, I think I missed this in my schooling, but can you talk more about this smoothie metaphor, please?
Yeah, you know what? You can tell who trained in biochemistry versus cell biology whenever you say cell smoothie by how hard they laugh / grimace. But basically, whenever we have systems that we're trying to create in vitro, it's a mixture of cells. So sometimes the way you get that mixture of cells is you go to an organ and you dissociate the cells, and you don't put it in a blender because that's when you go the biochemist route. But for the cell biologist in the room, we do tricks to dissociate the cells, and it just literally looks like a strawberry smoothie. That's what everyone's insides look like. Fun fact. Hope that doesn't ruin your morning routine or protein shake.
Okay, non-scientist here. That's a little gross.
Yeah, but it's totally utilitarian, right? So the whole point of this is that for whatever end application, we need to get to cells, right? But if we're harvesting them from whole organs, we have to be able to separate them and either purify certain cell populations, culture the ones we want, whatever your game is, you got to have them separated, and that requires a smoothie. I was really shocked to hear people even talk about optogenetics as a form of treatment. I'm familiar with that from neuroscience, where we're trying to use it as a tool to understand circuitry. How is this even an option for a therapy?
So optogenetics is absolutely fascinating. So it's the field where you're trying to make non-light sensing cells see light. And as you point out in neuroscience, it's cool. Like, can you make certain parts of your brain respond and see how it's connected? But in the eye, because you can actually do it in a live human person, maybe you can use it therapeutically. And so there's a lot of excitement in that area on seeing if once you've lost your light sensing cells, can you give that ability back to the second order neurons, which are these bipolar cells or the ganglion cells, actual cells connecting to the brain. Of course, you lose some resolution by doing that because there are about 200 million photoreceptors in our human eye, and you're down to a few thousand of the inner retinal neurons. But as I said, there is so much redundancy in our vision that there is a potential that these few cells could still give you some useful vision.
I saw at a conference a number of years ago, not that many years ago, I think some of the early testing of this kind of approach it's occurring in people who are fully blind, no light sensing whatsoever. And there was a woman that they tested this on who had no light sensation, and then they did this, and then she was able to basically locate a pen on a table where it was like high contrast, good light conditions, but like that is completely insane. Zero light sensation to I can find an object. And like when we're talking about being able to, I mean, the way that sight underpins your ability to participate in society.
Yeah.
It's not to say that you can't when you're fully blind, but we don't make it easy.
Yeah.
And to be able to find an object or even just detect, a little bit it's
huge.
Like science fiction, A of all, and B, just so massive an improvement for somebody's ability to like navigate or interact.
Yeah. And the field's moved quite a long ways. Initially, these optogenetic proteins people were putting had very small dynamic ranges, very small wavelengths of light they responded. But the field has moved so much now that the current clinical trials can actually work where you don't need special goggles to see that, things like that. They could work in normal light levels and within the normal light wavelengths. Of course, you won't get color vision back, but if you can even, as Susie pointed out, make sense, not hit a wall right in front of you because you couldn't see it, those are huge improvements in these patients.
And like as a person who, despite still being, I have good visual acuity on the eye chart, but like I run into things a lot. I get a lot of concussions. Knowing that like there may be, we can't fix all the problems, but like there is a possibility for giving back some light sensation. It gives me some kind of like, I don't know.
Gets you in the feels.
It really does. It really does.
It's the same kind of thing too, where you look at like how people have been kind of taking on like a different strategy for prosthetics. It's that same thing of like, how are you mapping into the brain and just restoring this? It's such a trip.
Oh, it's crazy. There was another podcast I listened to where they hijack other senses, and Deepak knows what I'm gonna say. There's this one episode where they like basically convert. There's a sensor that converts information about the spatial environment into electrical impulses, and they basically attach it wherever. But this one, it was to the tongue, so that you could see through your tongue, and it works.
The other science fiction thing that's happening is this chip technology. So there was this recent study which just came out. It's a company based on work here in Stanford of an electronic chip that has at least rescued some vision in patients. It's like a chip, it's about like 100 electrodes. You put it where your light sensing photoreceptors are, and the chip is wirelessly connected to a camera that you just wear as a part of your goggles, and that sends the signal to the eye.
What a trip.
So that was like one of those early things that I was working on prosthesis, and there was one company that made a little bit of a leap in that space, but again, it was just so difficult to get these chips to work. And this updated version of chip, the data looks really interesting.
Are they like carbon nanotube?
So, something. These are micro electrodes. They're spatially depolarized based on which area. And so originally when these were made, they were connected via wires to a sensor on the front of the eye, and that had to connect to a camera, and then a box that you would have to hang around and walk. But even those early studies were very fascinating. And there's videos of people who received some of these early chips. They could actually walk on the road because they could make out those edges of the roads, not fall down, see when to stop. And now it has moved to this new version of the chip, which is actually even better adaptable.
Well, so this brings me up to the next thing, thinking forward into the future. What are some things that you are the most hopeful for, or you think we're going to be like, just fast forward 25 years?
Oh my God, I really want a glaucoma drug that's not an eye drop. Like, desperately. Like, glaucoma, Deepak kind of mentioned it earlier, but like what you do when you get glaucoma is you put eye drops that sting into your eyes between 1 and 3 times a day. And that might help, but it might not. It's certainly not going to feel like it's helping. And same kind of deal where we're talking about like, oh, they're not complying with their treatment regimen. And I'm like, well, it hurts. And I can't tell that it's helping me.
And they can't see, and they're
And they
trying to
can't-
put these drops in your eye.
So I want like neuroprotective agents. So the eye drops, they're usually intraocular pressure lowering, which technically helps protect your neurons if there's too much pressure on them. But something that directly protects and supports the health of the neurons so they aren't dying, I would just be thrilled if we managed that.
For me, it's cell therapies.
Yeah.
I'm super excited about it. It's going to be a challenging ride there, but I'm very hopeful that's the only way we are actually going to rescue. Because you can do things to slow things down, but there are lots of patients who reach that stage that you have to figure out a way to rescue them. And to me, that's the ultimate rescue.
I have another one. I really.
What you got?
25 years. We're on the cusp of changing the way that we assess vision. So, like, the way that we do clinical trials now, by and large, for ophthalmology, is best corrected visual acuity, which is literally the eye chart. How many letters can you read? We talk about how the trial went in terms of how many letters you could read between, the group that was treated and the group that wasn't.
Does the Photoshop thing that you were talking about screw that up?
Yes, my best corrected visual acuity is amazing. And again, triangle, can't see Deepak. Like I have, that's not considered clinically significant. I'm missing over half of the vision in my right eye. It's significant, but it doesn't register on that eye chart. So there's other things like visual field, which there's, long established ways of measuring, but we're starting to see things like using virtual reality headsets, which as a patient, I can tell you, the test feels very similar, and they compare very well to the kind of gold standard way of doing this, Humphrey Field Analyzer. But I get to sit on my couch and I can do it, more often. It's so much more accessible. And that also gives us the ability in clinical trial design to actually take more measurements, which gives us more sensitivity. And you can use them to do other things completely, like mazes.
Or playing a game.
Or playing a game. My kids like it, yeah.
I know, I want to come over and play that.
There's a lot of cool stuff with visual endpoints, and that's just like tip of the iceberg. There's other kinds of ways to measure sensitivity, and just the idea of being able to measure vision in ways that are clinically meaningful and actually get at the, full, this is such a robust sense. We talked about how it's hard to know when it's changing. Being able to actually say, look, you have intermediate AMD, and even though you can't tell or you haven't noticed yet, like maybe it's just that your anxiety is a little higher and you don't know why, but it's actually because you can't see as well. Yeah. And it's just something we haven't been able to measure.
It's small changes, like you lose depth perception to some extent. All of that you really cannot measure.
Nighttime vision,
Nighttime
and like
vision,
suddenly you don't want to go out anymore, and you're like, oh, I just, don't really like it. And it's like, no, it's because you can't see as well.
What is your call to arms to the scientists out there? What breakthroughs need to happen in order to actually make that 25-year plan realistic? Call to arms, not a to-do list.
Yep. No, I'm thinking about my to-do list. It's a mixture of, bold science where we're, as individuals, as companies, as a field, we're willing to take smart risks on what we're trying, and really synthesizing what we understand across what different experiments are being done in the field. And then making small steps along the way at the same time.
That's what I was going to say. It'll be incremental. You don't expect it to be like hit the jackpot the first time, because we'll never do it. But using the knowledge that we're gaining to get that incremental success. There'll be obviously fairly large number of failures, but just accepting a failure and saying this is not going to work is not going to get us there. So we need to just be diligent at it, run the right type of experiments in the lab and those experiments in the clinic. And I think that's how we're going to achieve what we need to do, get to those patients eventually, because it's the patients in the end.
So like that, I'm at my core agree with y'all. Make those smart risks, take the incremental steps. But the other thing that sometimes whenever I'm talking to early in career scientists is sometimes surprising about what scientific backgrounds need to be at the table. So cell biologist, PK scientist, neuroscientist.
Yeah, I mean, there's a lot of what is necessary to move the needle on these different, potential transformations is super cross-functional, by which I mean we need a variety of backgrounds at the table. This isn't something that simply I don't know.
Single lab solving thing. It has to be a team behind. You need, depending on what you're trying to get, if you're looking at, say, optogenetics, you require how do you deliver it. So there's the virus people that need to work. Then there's the protein people to figure out how these proteins are going to respond to light. What's the right range we need to work on? There's people who know
structural biologists.
We need people with like engineering backgrounds.
Sure.
And we need people who can communicate between all of these different backgrounds, because a lot of us don't naturally understand what each other are saying.
Yeah, right? That everyone laughs when he brings stuff up like that because it's so painfully true.
It's super true.
And then if you're talking about like AMD or what AMD is, now you're looking at vascular disease, you have the inflammation disease. Now, not only that, we want a protein scientist who can connect those things together to make these bispecifics, multispecifics, hit multiple biologies simultaneously. That's what a multispecific is supposed to do. Reach the site of action. There's where the PKP people come, where it's like, no, we made this amazing molecule. It's so big that it just won't even reach where you want it to go. And
Bad news,
oh man, that's all. So
that's our backstory. That's our origin story.
Yeah, so it's one of those challenges that I think it's a team approach, and the more people talk to each other, I think that's the way to get there.
Thinking about optogenetics in particular, there are so many components to that you need to get to work. And how do you think about developing a therapy when you have to fix, 17 different things at once? And the answer is that you can't fix them all at once. You need to make progress on them individually if you can. And so having multiple kind of opportunities within one organization that inform on different parts of that equation can be super helpful, or looking, across the field at what are we learning from this attempt and this experiment over here, non-clinically or clinically. And I'm thinking about, there's been a lot of work on using gene therapy approaches for what's called biofactory approaches, where you're basically, injecting these into the eye and asking the cells to make a little something extra, maybe an anti-VEGF, for instance. And that's where a lot of the work in AAV or gene therapy in ophthalmology has been, not all of it, but using that to understand, how much are we getting in there and actually getting it to work and do the thing we're asking it to do can be really helpful when you're trying to figure out how to do something completely different like optogenetics, but maybe you're using the same way in, the same door. And then you're basically kind of simplifying that question a little bit by solving for something else in another location. And I like to see, particularly when I think about this from kind of a sort of organizational framework, how do the different things that we're pursuing inform each other and move us forward together instead of just thinking of them individually.
No, these cross group learnings are so critical as we want to move this forward.
That's the favorite thing about being a scientist.
It's so fun. It's so much fun.
I have to say, this has been a lot of fun. It was really great to meet you, Deepak, and it's so great to see you again, Susie. This was a lot of fun. Thank you so much for joining us.
Thank you for having us.
Yeah, this is awesome.
Danielle, really cool episode. Eye-opening, if you will. What do you think is the next biggest breakthrough in ophthalmology, given your experience in the field?
Probably the thing that I know the least about, which would be regenerative cell therapies. I think that's so amazing, so cool. And I really think that can get at some logistics that some of our classic pathways just can't even touch, right? Because like most of the time we're trying to like sop up a protein that's not supposed to be there or something like that. But having a therapy that can actually replace damaged cells, like that's groundbreaking.
It was incredible to hear about some of the technologies that take even non-light sensitive cells and make them quote unquote see. Do you think we're going to get to a place where we can really ameliorate the vascular diseases, or do you think we can actually go from zero and restore somebody's vision that never had it?
So I'd say both. We have strategies that are really great at dealing with the vascularization. We just need to improve maybe some delivery and make it easier for patients. But also, I think this idea of expanding how people can process vision, like that's something that's so new that we haven't been able to touch ever before. That I mean, that's really where it's at. I think that's cool. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stalking cells.